So a few things before we jump into today's lab. Like I mentioned before, there's a lot in today's lab, so we're going to be going pretty quick. Your lab final is next week. I hope that's not a surprise to you. All of your grades so far, with the exception of your lab final, will be on eLearn by this Friday. The reason that I'm not putting your work ethic and task points up until Friday is because I have to make sure you get your work ethic points this week. And then they'll be up. So by Friday, everything should be up on eLearn. If you want to know your task points for this lab, I can give those to you sometime today, but it's not going to include your work ethic score. Because that's where your work ethic will be taken from if you've lost work ethic points. Does that make sense? So on eLearn, if you've made all of the task points out of 14, but you've lost a work ethic point, it would say 13 out of 14. If you didn't lose any work ethic, it'll say 16 out of 14. So it's going to look like 2 extra credit points. Okay? Your lab final is at 11 next Tuesday. It's not an hour delayed or anything like that, like some of the other labs. It's designed to take about an hour and a half, and then you're done. I will have a survey for you guys after your lab final. If you don't do the survey, I won't grade your final until you do. So do the survey. It's anonymous, so it helps me in the future. Okay, one thing from last week that I believe I forgot to mention to you guys, from your dichotomous key, did we talk about the other clinically significant gram-positive cocci at the bottom of your dichotomous key? I didn't think so. I just wanted to mention them briefly. So you've got, mine says Enterobacter up here, yours says Enterococcus. Enterococcus is the right one, this is a typo, so just ignore that. Enterococcus is one that you sort of worry about a little bit. It can cause endocarditis, but also some GI issues occasionally. The bad thing about Enterococcus is that it has resistance. So it's at the very bottom here. Mine says Enterobacter. Yours says Enterococcus. Yeah, yours is right. Yeah. That's really all I wanted to say about that one. There's a drug called vancomycin, which is kind of like a drug they go to if you're allergic to penicillin or an antibiotic or a drug. Penicillin isn't working, so it's an organism resistant to penicillin. They'll go to vancomycin a lot, but we worry about vancomycin resistance with Enterococcus. Now, group B strep, that one you are going to see if you work in labor and delivery or in an OBGYN clinic. A lot of women, I think it's about 1 in 4 women carry it just as normal microbiota on their bodies, which it doesn't affect a female at all, an adult female, but it can affect a baby as it passes through the birth canal. So what they do is they check every pregnant woman that's getting prenatal care for group B strep. And it's called Streptococcus agalactiae, but everybody calls it group B strep because it's easier. They check them around 37 weeks or so to see if it's normal flora for them. If it is, when they go into labor, they're going to give them antibiotics to kill it off. Even though it's normal flora for mom, it's not good for the baby. So they'll just precautionary give them antibiotics just in case. It's really common. It's not, like if any of you guys ever have a baby and you have group B strep, it's not weird. It's not, I mean, it's super common. It's normal microbiota, which at this point, you know, we all have. But that one is one that they will look for. So I just wanted to throw that out there so that you're aware it exists. And you will probably see it at some point. If not working on labor and delivery, you will probably see it in a patient chart or something like that in medical history. Okay, how do you guys feel about the dichotomous key? Okay? Okay. Hopefully you'll know by next week. let's jump right into today's lab. So we've gone over several different ways that you can identify different organisms. The ones we mostly used during labs 10 and 11 with your unknowns were biochemical tests. So things like lactose and citrate. But today I'm going to show you a few different ones. There are antibody and antigen tests that you can do. So if you want to jump in your lab manuals, they are on page 72. Have you guys studied antigens and antibodies before? What shape is an antibody? Like, what does it look like? It's a Y shape. Okay, so we've got our antibodies here, these green Y-shaped things. Antibodies are what your body produces in response to a foreign substance, which would be an antigen. So let's say this right here is E. coli. On the surface of a bacterial cell, you're going to have some antigens. So you see here, you've got like a triangle antigen, a round antigen, a square antigen. Your body is going to produce antibodies specific for those antigens. So if you look closely, this antibody up here has the binding site is a triangle shape. So it's only going to bind to a triangle shaped antigen. It's very specific. Y'all with me? And this one over here is round. So it's only going to bind to a round antigen. So antibodies and antigens, or antibodies, are specific for antigens. I hope that this is just sort of like a quick review for you from previous labs. If not, we're not getting too in depth here. There are a few different tests that you can do. If you look in your lab manual, you can see them. So you can use antibody-based tests, which detect antibodies. So using antibodies to detect antibodies. And then you can use antibody-based tests, which detect antigens. So the first one I have listed here, you're looking for antibodies in the patient specimen. So remember, antibodies is what your body produces in response to a foreign substance like E. coli or something. If a patient has the antibodies for a specific organism or something like that, then they've been exposed to it at some point. It may not be there right now, like your example there, HIV screening tests. Like if you've been exposed to HIV at some point, you will have the antibodies for HIV, but the virus may not be present in very high levels at all. Y'all with me? Okay, so you're looking for the antibodies there. Now the problem with that is that you can't tell if it's a current infection. Okay, a lot of times though, you look for the antibodies because the antigens are not on the surface of whatever you're looking for, so it makes it a lot more difficult. So you would look for the antibodies. The other example would be Lyme disease. So they check these, or for your antibodies, for a bunch of different tests. So you've got HIV and Lyme disease listed there as well. Have any of you guys ever had something called a titer done? That's where they might be looking to see if you've been vaccinated for something. Generally speaking, they're looking for antibodies. In the hospital setting, you might see an order in the future like HBSA. Well, let's do HBAB. AB stands for antibody. HB is normally hepatitis B. So they're looking to see if you have antibodies to hepatitis B. So have you been exposed to hepatitis B, or if you got the vaccine, you would have produced the antibodies for hepatitis B. So if they're looking to see if you've been vaccinated, like maybe you lost your vaccine records or something like that, they could do a test like this to see if you've been exposed to it at some point. So you may see something like this, or HCAV, A, B, hepatitis C antibodies. I see it all the time in the lab. Just be aware what the AB stands for when you see it in the future. So tons of different tests. They're looking to see if you have the antibody. Now, there are some tests that use, that they do that detect the antigen instead. So they use antibodies in the test and see if the antigen is present. So a lot of times what they'll do is have an antibody. This picture up here is an example of the other one, but they'll have an antibody and they'll label the antibody with like some fluorescent something. And this is done like at the manufacturer. And then they'll put a patient sample in. And if that antibody binds with something in the patient sample, that means it bound with that specific antigen and it'll fluoresce. Something like that will happen and they'll know that the antigen was present. So they were using an antibody to find an antigen. Okay, so I'm going to skip back to HIV real quick because I'm going to show you a quick video. Here, some tests are done this way. It's sometimes called the sandwich assay. Assay is just a fancy word for test. You have some tests that you have like a little test tube and the antibody for whatever you're looking for, let's say HIV, is already made into the wall of the test tube. And then you add the patient sample to that test tube, and let's say the patient has whatever organism or virus you're looking for, that antigen is going to bind to the antibody that was made in the test tube. Y'all with me so far? But you can't tell yet until you add antibodies. So you add another group of antibodies. These are generally the same as the ones that were already added to the tube, except for that they're normally labeled with something, like labeled like a colored bead or like a fluorescent tag is how I kind of think about it. So that fluorescent tag is going to show a reaction or like a color, and you'll be able to see it, but only if it binds. Y'all with me? It's kind of confusing. That's how a lot of them work. So I'm going to show you a quick video on how an HIV test works, and then we'll talk about it.
So one of the tests.
Okay, so that is how an HIV test is done. Those are done pretty frequently in the hospital lab, not only on patients, but on any staff that maybe like stick themselves with a needle from a patient by accident or something like that. They'll have to do that right away, just to check for it. Any questions on that, how that one's done? Okay, so the next one I want to talk about is your strep test. Now, before we go into that, you guys are going to be doing a strep test. So I want you guys to turn on your microincinerators, but you don't have them at your tables. So you only need one per table. If you guys want to come grab like one per table, here's table 6. And go ahead and plug them in. There's 6. There's 1, 3, these aren't labeled, but you can use them. Oh wait, that one is. Who is that one?
4.
4. You can use this one. Which table are you? 1, 2. There you go. So go ahead and turn those on while I'm explaining how this works so that they can heat up a little bit. I'm going to explain it in my phrasing. You have the exact way that this test works on page 72 and 73. So if you see this or these steps, starting with strategy behind antibody-based antigen detection. So we're going to be using antibodies to find antigens. There's steps 1 through 6. Those are a lot of words. And then there's picture after it, which is this picture. I'm going to show it to you in my words, which is actually going to be very similar to these words, but I think it's a little bit easier to understand. And you don't necessarily have to be, you don't need to memorize this, I would say, but I just wanted to explain it to you because I think it's cool. So, how many of you have had a strep test done? How many of you have never had a strep test done? Oh, y'all are lucky. Okay. How this works, and this is how you guys are going to do it. You have a little test tube. It's tiny. It looks like this up here. It's a clear test tube. I have a piece of paper in it right now. And then you have 2 reagents. You drop 4 drops of each reagent into that little test tube. And then you mix in a patient throat swab. Okay, and that's how this sort of works. So, I have my little drawing up here of sort of the steps of what's happening when you're doing a strep test, okay? So you add in the drops of reagent. That reagent contains anti-strep antibodies from rabbits, okay? So they gave rabbits strep and then took their antibodies, is how that worked, okay? So these anti-strep antibodies are labeled with a colored bead. So really, this should look like the little red circle is a colored bead specific for those anti-strep antibodies. Y'all with me so far? So far, so good. They're from rabbits. You add your patient sample, so my lovely swab is right there. How'd they get the swab? Yep, it's just a throat swab. Hopefully they only had to get one. You mix your swab into that reagent, and what I have drawn up here is like this purplish color are strep antigens. So that's Streptococcus pyogenes from the throat swab. Okay, so I drew 3 of them. This anti-strep antibody is specific for strep antigens. So any strep antigens are going to bind to strep antibodies. Y'all with me so far? Okay, so we've got strep antibodies, and then I have 3 strep antigens, let's say. So they bind to 3 of the strep antibodies. So far, so good? Okay. Then you have a little sheet of paper that looks like this, and you've got a test kit. So this is all it looks like. It doesn't look fancy at all. Okay. And you stick it down into the tube and wait for 5 minutes. What that's going to do, you're not going to be able to see any of this going on, but this test strip has 2 stops of antibodies built into the paper, which is pretty cool. They did this like at the manufacturer. So you can see I drew 2 lines of antibodies. The first line is going to be the first spot where liquid is going to hit. So I'm saying first stop because it's the bottom one, which is kind of weird, but if you think about it, when you put paper into liquid, it's going to draw up. Y'all get to see this in a little bit. So the first one it's going to hit is this one, and you have a specific direction you have to put it in the liquid, so you can't really mess this up. So the first one you'll hit is this line. This line are anti-strep antibodies. Okay, so they built in anti-strep antibodies into this little paper. So as your liquid and antibodies and possibly antigens go up the paper, they're going to be drawn, and anything that's attached to a strep antigen is going to stop at the strep antibody line. Y'all with me so far, so good? So antigens can be attached to more than one antibody. So this little purple antigen is the strep from the patient's throat. It's attached to the anti-strep antibodies from the reagent, and then it's also going to attach to the anti-strep line here. And it's going to bind there. Now, anything that doesn't have strep is not going to bind there. So y'all see my one little antibody that didn't attach to strep, it went straight past that line. Y'all okay so far? Okay, so anything that's not attached to strep is going to just bypass that first stop and go up to the second one. That second line of antibodies are anti-rabbit antibodies, which is weird, but any rabbit is going to bind to this anti-rabbit antibodies, and if you remember, these were from rabbits. So anything in here that hasn't bound at that first line is gonna bind at that second line. Y'all with me? So any rabbit anything is gonna bind at this second line. That makes sure that you put in the reagent. So if you forgot the reagent, this line won't work, and then you'll know something was wrong. So if you see it's anti-rabbit, it bound up here. Y'all with me so far? Okay, now you're not gonna see any of that. What you're gonna see is 2 red lines because of those labeled antibodies at the very beginning. Okay, so if they're labeled red, you're gonna get 2 red lines. That's a positive. So T is for test, C is for control. So that first line is the test, do they have strep? That second line is, did you add all of the reagent right and is it working right? Y'all with me so far? Okay, so that's what a positive is going to look like. What would this be? Negative for what? Strep what? Pyogenes. Yes. Okay, and I know it's hard to see right there, so I'm going to draw one over here. What about this one? CT. You didn't do it right, possibly. It could be that the reagent's expired or something. It's not working anymore. You have to do it again. Okay, because you always have to get that control line. What about that? I don't know. Did we just forget to do it? Maybe we were doing 6 at a time and we skipped over number 5. It's possible. Very rare that that would happen. Or that could be a negative, but the control didn't work. So you would have to redo both of these either way. Y'all good on this? Okay. False negatives. When do you think you could see a false negative here? So something that's negative, but it should have come up positive. Any guesses?
It's too early to see antigen detected.
Yeah, maybe there was only like 1 antigen in their throat and they didn't swab that part of the throat. Maybe they swabbed your cheek by accident. Have any of you guys ever had them do 1 throat swab and then 5 minutes later they come in and do another one? That could be a couple of different things. 1, they swabbed the wrong part of your mouth and didn't want to admit it right away. I'm just being honest. 2, they got a result like one of those 2 where the control line didn't work. 3, they got a negative and they really think that you have strep, so they want to swab again just to make sure. Maybe try like the right side of the throat instead of the left or something like that, whatever they didn't do the first time. Now, false positives are super rare. However, in my little medical lab community Facebook groups that I'm in, I have seen several people report false positives from eating flaming Cheetos. Fun fact. I don't know why. It's very weird. But they have like a correlation there. Patients that ate flaming Cheetos right before they got a strep test sometimes got a false positive. I don't know. Okay, so if you guys are comfortable with this, we're going to do it. You each have a tube at your table, an unknown tube, and you have a little card here that tells you exactly how this is done. So what you're going to do is add 4 drops of reagent A, 4 drops of reagent B into that little test tube. And then you're going to add your swab to the tube. So how do we get the swab? You're not going to throat swab each other today. What you're going to do is swab what's in this liquid instead. So you're opening a test tube. So what do you need to do with it? Run it by the micro incinerator after you open it and before you close it. Then you're going to stick the swab down in the tube after you open it. Press the swab to the side of the tube before you pull it out so that it's not dripping. And then mix it into that reagent really well. If you forget any of these steps, it's on the little card. So you're going to rotate the swab 10 times to mix it. Then you're going to wait for about a minute. Then you're going to take the swab out and put the test strip in the tube. Now your test strips have arrows on one end that point towards the end of the strip. This one at the very bottom, it has arrows that point one direction. They need to be pointing down. That's the direction you're putting the tube or putting the strip into the tube. Y'all with me? So make sure those arrows are pointing down. And then you're going to time it for 5 minutes. Now, if it's positive after 1 minute, you can be pretty certain it's positive. It's not going to change back. But if it's negative, you need to wait the full 5 minutes. Okay? So y'all go ahead and get started on that. You need to be wearing gloves. And you also should be wearing goggles if you're working with this bacteria. So go ahead and grab goggles out of the back.
The shrimp AG, that stands for what?
Antigen.
Okay.
On this side should have gotten negatives. Y'all should have gotten positives. Now I will say, if you want to view each other's, you can. Take pictures if you want. Did you all see the plates that I passed around? Okay. If you don't want those, everything can go in the red biohazard bags. Everything in the little tube. So just throw the whole tube in the little biohazard bags. And then get rid of your gloves. You don't need your goggles anymore either. If you want to go ahead and put those up. Okay, how do you guys feel about the strep test? Did you feel okay about it? Okay, so up here you've got a good picture example of a positive versus a negative. If I showed you this strep test, the organism that caused this result has what kind of catalase reaction?
No, negative.
Negative. Right, because it's strep. Yeah. The organism that caused this reaction on the strip has what kind of catalase reaction? Negative, because it's strep pyogenes. What would this one look like on MSA agar, mannitol salt agar? Right, it wouldn't grow. What's the hemolysis on blood agar? Beta, yeah, good. Okay, now some of you guys have been asking me about a faint line. Any line at all is positive. This test, the way that this one is done, is the same way that pregnancy tests are done. It's the same way that flu tests are done. With flu, it is slightly different. They have 3 lines, so I know this is going to be a little dark. But they'll have a control and then an A and a B. So they'll be testing for flu A and flu B. And then it'll go to the control. So it'll pass the flu A antibodies. If it doesn't have flu A, it'll pass flu B if it doesn't have flu B, and then it'll go to the control line, which everything will bind to. If they have flu A, it'll bind here, and then it'll pass B and bind at C. If they have flu A and B, they'll bind at all 3. If they only have flu B, it'll only bind here and at the control. So it's pretty cool. It's done the same way. A lot of hospital labs have a machine actually that they'll stick the strip into, and the machine reads it for you. So that prevents you from missing a very faint line. Have any of you guys heard somebody say like, oh, I took a pregnancy test and it was really, really faint. I don't know, should I do another one? A faint line is a positive. I know when I was pregnant, the line was like super duper faint. And in the lab, any line is a positive. So I have an example of a super faint line that is a positive that I'm going to pass around for you. And actually, I'll turn on the lights while I pass this around. For pregnancy tests, I will go ahead and tell you on an exam, I would not give you something like this because it's hard. But I want you to see there are faint lines that are positive. On a pregnancy test, instead of looking for the strep pyogenes, they're looking for beta HCG, which is just a hormone that you'll produce in higher levels if you're pregnant. And then flu is the same way. How do you guys feel about this?
The flu is the same way as, it's doing antigen testing, correct?
Right.
Not hormone.
Right. Yeah. Flu is looking for the flu virus. So antigens. Nose normally high up there, yeah. I hear they're not very comfortable. I've never had one, I don't think. Yeah.
Okay, so when we did the test, there's antibodies in the liquid that
Right.
bind with the antigen, our antibodies on the test strip. Like they'll bind to the antigens in the liquid, the antibodies will,
Right.
and then they'll bind again on the test strip.
Right. So those antigens can bind with more than one antibody at a time.
I think that's what.
Yeah, it's weird.
So you're looking for the antigen.
Right.
Okay.
Right. Using antibodies. Yeah.
So is that the same for the virus though, or is the virus
I know. Do
the
it.
same for the virus though? Because you're still looking for antibodies.
You'll still look for the antigens or antibodies. It just depends which one you're looking for. Yeah. For the strip like that, you're looking for the antigen. So you'd be looking for the virus
You're looking for the antigen, but you're using the antibodies.
to find it. Right.
The virus, you're using the virus.
Antibodies to find it. Right. Exactly. Yeah. Okay. Can y'all see my very faint positive? Yeah, this is like a constant thing in here. Yeah, really? I'm surprised, honestly. I'm glad this is the first time y'all have seen it. Okay, so how do you guys feel about those as far as like identifying a positive or negative on an exam? And that strep test is looking for what? Streptococcus pyogenes, which causes what? Yeah, good. Okay. We're going to move on to a different type of test. So you've got biochemical tests, you have antigen, antibody-based tests, and then now I want to talk about molecular testing. So sometimes they use nucleic acids to identify certain organisms. This one's pretty cool. So how this one's done, it's actually listed pretty well in your lab manual on 75. What they do is first they had to have a patient sample, and they collect the patient sample and then treat it so that you can get DNA or RNA from the organism that's in the patient sample. Okay. And then if it's DNA, you have to separate the strands. So you need a single strand of DNA or RNA. Then you'll add a label to a single-stranded nucleic acid. So a label is normally something that's just going to fluoresce or cause a color change or something like that. And then those are put onto a test platform. So you have a test platform, which is basically just like a piece of plastic with a bunch of different tiny little holes in it that are sort of like test tubes, not actually holes, but they're like test tubes that are really, really small, like about this deep, okay? And it looks kind of like this. And in each one of these, they have put a single strand of a different organism. You're going to add your patient sample to each of these little test tube looking things, and then you wash it off, okay? If you have a single strand match up with a single strand from the patient, so like this one, for example, let's say this one is a single strand of hepatitis A. This one is hepatitis B, hepatitis C, okay? And you put your patient sample in each one. If you have a single strand of hepatitis A in the patient sample, it's going to bind with that single strand of hepatitis A in the test tube, and when you wash it all off, it won't wash off if it's bound together. But if it's only one single strand, it's going to wash right out if nothing has bound together. So it's kind of cool. Y'all with me? And then if it is bound together, it's going to fluoresce. So you can see these different ones are bright green, so they've fluoresced, so that patient has the matching strand for whatever organism is in that well. So it's kind of cool. Do you guys remember the stool samples with Giardia in the past and how hard it was to see Giardia? I use this for Giardia in the lab. It's much easier. You don't miss it nearly as often. So you're looking for a DNA strand of Giardia is going to be in here. You're going to get a patient sample, put it in this well, and then if it fluoresces after you wash it off, then Giardia is present in that patient sample. So it's kind of cool. Yeah.
So is the pairs like made into the tray?
Yeah. Yeah. So like the manufacturer will put the strands in the tray before you even put the patient sample in there. So it's pretty cool. This is another easier picture to see to me. Did you want a picture? I think the other one is easier to see, honestly. So you've got DNA strands in every little block. And then if your patient matches one of these DNA strands, or their blood or something has some of those DNA strands in it, they're going to bind together, and then they'll fluoresce. If they don't bind together with anything, the patient sample will just wash right out. So it's pretty cool. Okay, so we can't do this in here. It takes a little while, but we have a good simulation for you. At your tables, you have a probe platform. And what you're going to do with this, actually, let me have this up. Okay, what you're going to do with this is like your little well of DNA strands. So you guys have the platform. I have the DNA strands that are going to be put in to the wells by the manufacturers. Okay, those are called probes. So these probes you're going to stick on. I'm going to give you some. They're labeled like probe 4. So probe 4 is a DNA strand, and you're going to put it on probe 4's spot on the platform. Y'all with me? Okay, so you're going to go ahead and put all of the probes on, and then you have DNA or RNA. I don't know which one you'll get, maybe both. And you have to compare the strands on the probes to your patient sample. So your patient sample is labeled with a fluorescent tag, and it's a DNA strand that you have to match up. Do you guys remember your base pairs? Mm-hmm. What goes together? C's and G's go together. A pairs with T. C pairs with G. What about if you have a U? A. Okay, so you may get RNA, I don't know. So then once you have all of yours matched, I will just go ahead and tell you, you're not going to have a patient that matches all of these. You should have at least one, because there's a control built in. So number 1, probe 1, should match with something because that's our control. So any human DNA should match with this strand. Okay? Once you have them clipped together, so like say this matches probe 1, just clip it on there where the paper clip is. And then anything that isn't matched up with a fluorescent tag, you wash off. And anything with a fluorescent tag is what your patient has. So if you have probe 1, our positive control, and probe 4, your patient has malaria. Y'all with me? So that's how this works, okay? So I'm gonna pass these around to you and work on it together. Get comfortable with it. Good luck. Y'all will do just fine. And once you have your disease, I guess, let me know what you think it is. How did you guys feel about that? So that's pretty cool. It's quick. They also use this for a lot of STD testing. So some organisms are difficult to culture, so they'll do this for those. So a lot of places will use this for gonorrhea because remember, gonorrhea dies really easily. And they'll also use it for syphilis as well. A lot of times they actually have like entire plates that are just STDs, which is pretty cool and quick. Okay, so we're gonna switch gears here and start an activity that is a little bit different. We've been through all the testing that we're gonna talk about for the day and really for the semester. So this is gonna be kind of applying all of it together. And then we're gonna do an activity. I'm gonna go ahead and show this short intro video and then we'll do the activity and I'll explain it. I have a few more things after this.
In a hospital boardroom.
Okay, so the activity is going to be getting one of my laptops, or if you have a laptop, you can use yours. If you have headphones, I have a headphone splitter up here if you want to stay in here. However, I know a lot of you probably forgot headphones, which is fine. As long as you guys aren't all in this lab, I think it'll be fine. So you can take your laptop and go down to a booth down there or something like that and all listen to it together. What you do is click on your character. So I'm going to give you guys a different character. So if I give you Kelly, you'll go through this and when you pull it up, it's a CD. You're going to click skip the intro. And then you'll click the character I assign you. So if you got Kelly, it'll start playing Kelly's story. And then at the end of 58 seconds,
You got up.
it's going to ask you, like, it'll give you a choice of something to do. It'll say, like, should you do this? Should you do this? Should you do this? And there are wrong answers. And people might get upset with you if you choose the wrong one from experience. So go through this and then meet back in here. I have a sheet of paper for you to fill out while you're working on it. So here's your sheet of paper, one per table. Just write all your names on it. I want tables 1 and 2. You guys go back and be Kelly. These two go back and be Dina. So Dina is the nurse. This one's Dina. Okay, and then the back 2 tables, I want you guys to go back and be Manuel. Okay? So grab a laptop if you need one over here, and a CD and a sheet of paper. And y'all can go anywhere. Just don't go into a classroom where they're teaching, obviously. Just one sheet of paper. Yeah, you can tear into it if it's not open yet. Okay, so what we're going to do is I want you guys to just tell me something about your character that the rest of the class wouldn't know unless they watch your character's little video. So something you learned about the character that may have explained the reasons they were acting like they were acting, okay? So if you guys want to go first. You guys had Kelly, right? So Kelly was the, so that kind of gives you a good idea, like you don't really know what people are going through, patients along with family members and the staff. So I want to play this little video for you. It's short. I watched this video when I was going through orientation when I worked at Duke, so when I first started working there, and it has stuck with me ever since I watched it. It's a really good one, and I hope that it sticks with you like it stuck with me. So I will turn this on and play it because it's really good. So you guys can probably see why I like that video. So I hope that that helps you bring it all together. This is going to be y'all very soon, if it's not you already. So first you have to take your Microlab final. So that's next week. I'm going to do a quick review with you guys. First I want to show you on eLearn. Have I shown you guys those PowerPoints that you can complete? I showed you the first week, I know. Did I bring it up last week again? There are PowerPoints for you guys to complete if you want to under course content. They're on the left here. Do y'all remember this? It's like a little template where you put in your picture and a description. I haven't talked about it since first lab. For labs 8 through 12, once you get it to load, it's loading. You can download it, and then when you scroll down to lab 11, you've got pictures on here already dropped in. So if you want pictures, you can look at those there. And it even says like Serratia, Proteus. It says the organism names, and then you've got like Kirby Bauer, catalase, coagulase, gram stain pictures, things like that. So I would suggest that you use this. I think it would help you. And then you've got gram-positive augers as well. Again, though, with blood auger, it doesn't really help you to see the picture, in my opinion, in MSA. So I just wanted to point that out to you. You've got some streps on here, too, in case you weren't aware and haven't been using that for this semester. So I have my little final review. It's quick, just like the midterm one. So I'm going to go through it. If you guys remember, your final is worth 50 points. So you can all get a 50 out of 50. I would be super excited. You are going to have 49 questions. So it's 50 points. One is just worth 2 points. It's going to be set up exactly like the midterm was, with some questions on paper, some around the room. You can go in whatever order you want. 10 are going to be not multiple choice. So they're like short answer, fill in the blank, just like the midterm questions were. Okay, any questions about that? What time does it start? Yes. Okay, so what did we do? We learned how to make a slide. Y'all are experts at that at this point. You learned about stains, basic versus acidic, different types of stains. You learned about different staining procedures. So You've got your simple stain where we actually used methylene blue. What does a simple stain do? It's one basic stain. What can you tell by it? You did your negative stain with the wet mount. That is clinically significant because of Cryptococcus neoformans. So that's the one that is done all the time in the clinical lab on spinal fluids. You guys use Saccharomyces cerevisiae. That's the one where you could see the yeast budding, if you remember. The background was black because we used India ink, which is an acidic stain, so staining the background. I hope this is ringing a bell. That's kind of what it looked like. We did a capsule stain where your cells should have been purple and the background should have been black. I know for some of you guys, the cells didn't turn out as purple. So if I were you and that was the case, maybe look at some pictures. There are capsule stain pictures on eLearn. How did you make the capsule stain? You used that spreader slide, if you remember. Which slides did you have to heat fix? For your lab, the entire semester, you heat fixed all but one, right? Which one did we not heat fix? The wet mount. Yeah, so that might help you remember. Only one that we didn't heat fix, and that's because all of the other ones had a rinsing step. So we rinsed it off. We didn't want to rinse off our organisms. So what does heat fixing do? Hopefully you guys will remember that. It makes the organism stick to the slide, and it also kills the organism. That was your capsule stain. The capsule doesn't stain because it's neutrally charged. Gram positives and gram negatives. When we do the gram stain, what is differentiating between the two? Like, what structure are we really looking at? Yeah, the peptidoglycan layer of the cell wall is really what makes a big difference. Know the steps of the gram stain, what they look like after every step. What is the counter stain? It's cut off here. What's our good? Yeah. What color are gram positives? What color are gram negatives? Pink. Yep. How would you report them to a physician? You report what you see. Don't guess if you're unsure.
Is it better to be more vague and like?
I mean, if you can tell for sure, put it. But if you can't tell for sure, don't put it. Like, if all of the organisms are in clumps together, put that they're in clusters. If all of them are in chains, put that they're in chains. But if some look like they're in clusters and some are in chains, just don't put anything. Or you could put both. Gram negatives, we don't normally report the arrangement of. It's a lot of times it's difficult to tell. How would you report that one? Gram-positive cocci and chains, or you could put gram-positive cocci and chains and pairs, because I see little pairs up here.
That would be, that spot on the left over there.
Here?
Yeah, that's not a cluster.
See, that's where if you put it, I would take it, because I see it too. Yeah. I mean, if all you saw was like just this one and you put a cluster, I would mark that wrong. But if you're seeing what I'm seeing, or if I can see where you saw what you wrote down, I'll give it to you. I'm not that mean.
So we're doing this on the exam, like identify.
You probably will have to do this on the exam. What about this one? Gram-positive rods. Gram-negative rods. Gram-negative cocci. You guys also did an acid-fast stain. That's the one where we used a hot plate and little aluminum weigh boats where we had to heat it up before we put the stain on to allow the stain to kind of melt the cell wall a little bit because of that waxy layer. What was the primary stain here? Ooh, that's a hard one. Carbon fusion. Yep. And the counter stain was methylene blue. Decolorizer was acid alcohol, not acetone alcohol. You're going to get a pink or blue, or a reddish pink or blue. Again, this picture is not very good. If I put a picture on the exam, it would be much more clear than that. You would also have to know which staining procedure I used. So the organism we look for on an acid-fast stain is normally Mycobacterium. They would not gram stain. So Mycobacterium tuberculosis is that organism. You did the endospore stain. You also used heat to dry the stain through the endospore wall. What organism has endospores that we talked about a lot? Clostridium, that's true. And Bacillus. Yeah, those are two biggies. Well, we haven't really talked about Clostridium as much in lab, so if you didn't know that, that's not a big deal. Don't worry about it as much. But you do need to know Bacillus has endospores. That's what it looks like. Malachite green and saffronin. Oh, for this one, for those of you on my Instagram account, I was disappointed because I asked, what's the counter stain? And over 50% got it wrong for this one. What's the counter stain? Saffronin. I had a lot of people put malachite green. So that's the primary stain. So you'll want to know the difference between the two. Specimen collection, what should not contain normal microbiota, what should. Normal flora expected, throat, sputum, fecal, surface wounds, should be sterile, any body fluids, and deep wounds. Any body fluids, including urine. We talked about antimicrobials. Remember, you only need to know the ones that are bolded and italicized in your lab manual. Know their mechanism of action. So if I ask, what's the mechanism of action of Cipro, you should know it inhibits nucleic acid synthesis. So there are 5 of those I want you to know. You did this test. What is it called? Kirby-Bauer. What's the auger we used? Mueller-Hinton. Is it ringing a bell? What did we measure for the zone of inhibition? So that zone of inhibition is the area where bacteria doesn't grow. What did we measure? The radius or the diameter?
That was the radius, because it was from the medicine to the, I don't.
I am hearing mixed answers here. Who's confident? Like for sure, 100%. It's the diameter. Diameter. If you don't have a zone, you measure just the disc, which is about 6 or 7. You measure the area closest to the disc all the way across to the area on the other side closest to the disc. So like this one looks kind of like it has 2 zones. I would start it here instead of out here. Okay, if I did ask you to measure that and apply it to the chart, I would have to give you the chart as well. Know it. The dichotomous key. I would look at this, have it be the last thing you look at before you come in, and the first thing you write down when you get your exam. That's my advice to you. Again, I would abbreviate stuff like, oh, and look, that's actually flipped, so that's confusing. Like CIT plus KP. As long as you know KP is Klebsiella pneumoniae. That might make it easier. Come up with a little phrase or something that matches those letters. I don't know. Okay, then you have your gram-positives with mannitol salt agar, which is both selective and differential. Selective because of the salt, differential because of the mannitol. Staph aureus is yellow on it. Staph epi doesn't change the color. It's kind of still like a pinkish color. Sheep blood agar. There are 3 types of hemolysis. What do they mean? Which ones are which? Which one's alpha hemolytic? Partially hemolyzed. What organism exhibits that? There's only one. Strep pneumoniae. Yep. What about gamma? Gamma is no hemolysis. Staph epi is gamma. Alpha is that light gross green color. Gamma is no hemolysis, and beta is full hemolysis around the colonies. McConkey are either going to be, you're looking at the color of the colonies, not the color of the agar. They're either going to be pink or like a nudish color. Pink means they ferment lactose. Nude means they don't. Citrate, it's either going to be blue or green. Remember, if it's like a greenish blue and not like the blue of your trays, then it's negative. But if it's like the blue of your trays, it's positive. If you are doubting the color, ask me. I'll tell you. Well, you have to say, this is blue, right? And I'll say, no. It's not. Okay? Or this is pink, right? No, it's not. Yes, it is pink. I will make sure it'll be obvious for you guys, though. I'm not trying to trick you. I promise. We did API tests. If I gave you an API test on the exam, you would have to have all the information that I gave you before. So, what's a positive, what's a negative, the little code chart.
So you'd give us the chart and we.
Yeah, you would have to.
Organism.
Yeah, you would have to have everything. You would have to have a little score sheet. Positives, you add up. Negatives, you don't. You add up the little number underneath that box on the score sheet. Catalase test. Positive is staph, negative strep. Which one do we do a coagulase for? Positives only. What is a positive coagulase? And actually, that's not a good picture. It doesn't really look like that. But y'all saw it, right? The one that's clotted at the bottom is positive. Which one would that be? Staph aureus. A negative coagulase would be what? Staph epi. Yep. Strep pyogenes. Positive 2 lines, negative 1 line. You always have to have at least 1 line or else the test didn't work. We did the little nucleic acid simulation with the probes and the DNA strands. Any questions?
Will we have access to this project?
No. Sorry. Some of the images are copyrighted, so I can't put them online. That's the reason why. Yes.
Like after you said this, but is it just, is that like 8 through 12?
Oh, good question. Okay, so I could ask you anything from the entire lab manual if I wanted to. However, I don't really like to say it's cumulative because I'm only going to ask you stuff that you needed to know for the second half. Like, for example, if we have not ever mentioned it a second time in the second half, I wouldn't worry about it. If we have mentioned it again and maybe like you had to do it, I would know it. For example, you had to do a streak plate in the second half for your unknowns. I taught you how to do a streak plate in the first half. So you should know how to do a streak plate, even though I taught you in the first half. So if you wanted to revisit it, it's in the first half. I taught you about selective versus differential media. That was in the first half, but you had to know it for like MSA, and blood agar and citrate. I taught you how to use a microscope in the first half, but you used it in almost every lab in the second half. So you should probably know how to use a microscope on the final. Does that make sense? Like if I didn't mention anything about it again and we never ever talked about it again, don't stress over it. Does that help? I mean, it could be on there, but I'm only asking 49 questions. So I wouldn't stress about it.
How many are
Like a practical station? Probably like 15. Okay, I have one video before you leave. Do you guys like Frozen, the movie? This is a parody, so don't hate me if you don't like it. These people are awesome. If you're struggling with something, look up these parodies because I don't know why, but there are a lot of them online and they're funny. And if you can remember song lyrics, you might be able to remember some stuff that's going to be on the exam. So I'm going to play this little video. It's short. And then you guys are free to go. If you want to know your task points, just come up here and ask me after, and I'll tell you what they are.